friends, so this is my first experience of showing you something like this in YouTube. So I think you would like it. You would like my teaching, you would like my slides. If you like it, please like it in YouTube and subscribe it. Please, it's a hearty request. Now, today we'll talk about cells and organelles, chapter 1. That's topic 1, you can say. In this, we will generally talk about the outer membrane of the cell. All the outer membranes of the cell, we'll talk about that only. Next, more topics, we will talk about more about this topic, or other videos we can talk about other subjects that depend. I will now upload this type of videos only. Now, what is cell? We must know for that first. Cell is the structural and functional unit of life. It is a basic structural and functional unit of life. I repeat again, cell is the basic structural and functional unit of life. Our body, our whole body is made up of cells, small cells. The cells were first discovered by Robert Hooke in 1665. I think you all know that. But cell theory is something different, produced by Matthew J. Sladen and Theodore Swan. Also, Rudolf Virchow made some efforts. That is different. That were the postulates regarding to the cell. But when the cell was discovered by Robert Hooke in 1665, all right. So, through the slides, we'll try to understand few topics about the outer membranes. My name is Akash. Okay. So this is the pyramid of life. This is a pyramid of life. You can say that is in the form of pyramid. This the thing is very simple. You all know that atoms and molecules. This both can be the particles, smallest particles of matter. But also this can be that two atoms form one molecule and many molecules form one matter. That also can be possible. Or one molecule can make an matter. Many molecules can make an atom. Many molecules can make a matter, or many atoms can make matter. Same thing. Just these are two different identities, or you can say the smallest particles of matter. That's it. Next comes the organelle. The organelle is a matter, you can say. Okay, organelle is a matter. How organelle is a matter? Organelle is a matter because it is comprised of the atoms and molecules. You can say that organelle is a matter because it is comprised of atoms and molecules. Now, different organelles in the cell also. Lysosome is there, mitochondria is there, cell membrane is there, many more. Vacuoles, Golgi bodies, ER, endoplasmic reticulum, all are organelles. So, cell is comprised of organelles. There is something missing here. I think you all can identify that after cell there should be one more stage, one more pyramid step, that is my handwriting is not too good, but it's my best try. Tissues. It is the tissues. Many cells, many small cells, they combine to form tissues, and many tissues form organs. For example, meristematic tissue is a tissue. Then epithelial tissue, epidermal tissue, skin epithelial tissue. In plants, it is epidermal tissue, this outer layering. But meristematic tissue, there is something extra. I want to say it now only that meristematic tissue is the tissue which helps in growth of the plant. It is present in those plants which are growing, not in mature plants. It present in those plants which are growing. Okay, so many tissues form organs. Now many tissues form organs. Organs example that can be the lungs, the liver, the kidneys, the stomach, the nostrils also, windpipe, all are organs. Heart, brain. Brain is called the master organ because it controls the whole body. So many cells form tissues, many tissues form organs, and many organs form organ system. Like the nostrils, the windpipe, and the lungs, they form the respiratory system. The kidneys, the stomach, the liver, the small intestine, large intestine, all they form the esophagus. All they form the digestive system. Clear? Now the organ systems form an organism. In our body, many organ systems are there, especially the respiratory system, the digestive system, the muscular system, the nervous system, circulatory system. All are different type of organ systems. The formation of organs in a particular way to form a particular type of function, that is called organ system. Okay. Now many organisms have some population. Many organisms, if in a colony they live, that can be called population because they are populating more in number. They are more in number. Not only one organism, they are more in number. So from population they become community. A certain community, like in India. Let us talk about human organism. Human organism, if your own colony, if you see, if you suppose you live in Gujarat, suppose you live in Gujarat, and suppose you live in a place in Gujarat where only Gujaratis are there, no one else, only Gujaratis. So you are an organism with population, your own colony, just a colony, that is your population. That total can be called a population. But the whole city where only Gujaratis are present, same type of organisms, that will can be called a community. And many communities like that, one city, they form a state, that is the ecosystem. And the states may form many groups of states may form a biome. Many groups of ecosystems form a biome. So many states are there. States, some states, group of some states, suppose Gujarat, Rajasthan, Madhya Pradesh, they all will combine to form the biome. And groups, suppose Gujarati group is this, Rajasthan, Gujarat, Madhya Pradesh, suppose Bengali group is West Bengal, Bihar, West Bengal, Jharkhand, Orissa, suppose South Indian group, Karnataka, Tamil Nadu, Kerala, like this. They form a country, they form India. That India can be called a biosphere. Am I clear? I think it is very much clear to you. Now, this is all about the pyramid of life. I'm really sorry. Yes. So, plasma lemma. Now, I'll talk about plasma lemma. Now, what is plasma lemma? Plasma lemma is the other name for plasma membrane, or if you are communicable with, more communicatable with cell membrane. That's it. Cell membrane can be also called plasma membrane, also called plasma lemma. All are same. Clear? It is the outer covering of a cell. All cells they have, whether it's a plant cell, animal cell. Animal cell is the final covering. Plant cell, and we'll talk about some other types of cell. They will have an extra covering, that is called the cell wall. We'll talk about that. So let us consider plasma lemma. See what is written here. The cell membrane or plasma lemma is an universal system. What is the meaning of universal system? That means all cells are having, whatever may be the cell, prokaryotic, eukaryotic, plant cell, animal cell, fungi cell, bacteria cell, everything they are having a cell membrane. If cell membrane is not there, that cannot be called a cell. Clear? So universal system which form many structures of a cell. Each cell is bounded by a cell. The plasma lemma. Sorry, it is wrong given here. It should be each cell is bounded by a cell membrane. Okay, you all can understand. Should be cell membrane. Membrane. Cell membrane. Cell membrane, whatever. You can understand. So the plasma lemma or the cell membrane includes three layers. Three layers. Plasma lemma is outer covering, but it has also three layers. How? The outermost layering is applicable to the atmosphere. That is called the glycocalyx. Outermost covering, this covering is the glycocalyx. It is comprising of carbohydrates. This we will require, so you need to keep in mind that it contains carbohydrates. Glycocalyx is the outer covering, it contains carbohydrates because glyco meaning glucose. Glucose is a sugar, sugar is the carbohydrate. Always remember this derivation. Glycocalyx, glyco meaning glucose, glucose is a sugar, sugar is a saccharide, you can say, and saccharide always it is a carbohydrate. There are three types of carbohydrates, saccharides, different types of saccharides, monosaccharide, disaccharide, polysaccharide. It is a polysaccharide. The cell membrane is made up of polysaccharide carbohydrate. Remember this, just polysaccharide carbohydrate. Achha. Lipid bilayer. What is lipid bilayer? You must understand this concept. Lipid bilayer. Lipid means fat. Here you can say lipid molecule and proteins. Two things are there. That's why it is called bilayer. Lipid bilayer. Also, lipid is a molecule. So molecule, I said, it can be single or it can be two atoms also. So lipid molecule, lipid bilayer. So lipid, here it is two atoms, or you can consider molecule, that's same. So two layers are there combined in single layer. So that's why it is called bilayer. Two things are there. Lipid also there, proteins also there. Now this last layer, that is glycocalyx, lipid bilayer. I'm sorry. Some and last one is near the cell in contact with the cytoplasm. The glycocalyx is contact with the outer part, but the submembrane cortical layer that is in contact with the cytoplasm, you can see the liquid part of the cell. So it contains actin microfibrillans. Actually, it's filaments. We can also call it fibrillans. Okay, let us consider filaments. Actin microfilaments and microtubules. Actin microfilaments and microtubules. Now what is this? Actin is a type of substance or chemical, whatever chemical substance, which helps to protect the main part of the cell. Main part of the cell, or the transport materials through the cell. Let me make you understand. Suppose this is a cell, this is a cell, and this is a cell. Plasma membrane, one more thing I want to say, plasma membrane is always selectively permeable. Now, what do you mean by selectively permeable? It allows selected materials to pass through it. Okay, allows selected materials to pass through it. So these two are cells. So okay, selectively permeable. But how do it will enter? There is a membrane here. There is totally a membrane. How it will enter? Let it be selective permeable, but it just to open. It cannot go inside. You will read about the cell wall. It is rigid. It will not open only. It may open. So these openings, the microfilaments and microtubules, when they open to take in different molecules or different materials, different matter like sodium, potassium, when they open to take them, that that layer helps to form the openings. Submembrane cortical layer. That's why it is called submembrane, so that it can open to take in the things. It opens the lipid bilayer, and through it the materials come in. Again, when the material transfer is stopped, closed. Clear? Now this is the structure of plasma lemma. Okay, this is structure of plasma lemma. Plasma lemma is a single thing. You can understand how minute it is. The study is how minute. Cell only we cannot observe. Cell what? We cannot observe tissues even. Organs we can observe, but we cannot even observe tissues. And also some tissues, larger tissues we can observe. But cells, very rare cells we can observe. Yes, you can talk about the algae cells. That's all right. The spirogyna is all right. We can. It is a very long thing. We can easily see. Algae is considered a cell, multicellular organism, but it is comprised of cells. No, we can understand the different filaments that are only the cells, but very rarely we can see the cells. And that cell has some organelles, which is one is plasma membrane, and that has some structure, such big structure you can see here. So that is very inculcative thing. The thing is very inculcative. So you must understand this concept very clearly. So plasma membrane structural components. Uh, This is a fluid mosaic membrane model. Fluid mosaic means fluid and mosaic. These two things were taken together to depict this. Actually, this is not actual thing. This is just a depiction. Yes, to understand the structure. This was made by singer Nicholson, not the singing a song. That not singer. No, I'm not talking about. I'm talking about the name. It was named singer Nicholson. I don't know why, but fluid mosaic membrane model. This model was made by singer Nicholson. You must remember it. Singer Nicholson. Just okay. So you can see here different proteins are there. Plasma membrane is consisting of carbohydrates, the glycocalyx layer, upper portion. Upper portion. You can see the most upper portion only glycoprotein is there. Blue color. Blue color thing, that is the, this one is the glycoprotein. Right. This one is the glycoprotein. The blue color. So glyco meaning, I said glycocalyx that time. Glyco means glucose. Glucose is a sugar. Sugar is a polysaccharide. Polysaccharide are carbohydrates. So it is carbohydrates. So glycocalyx, that means the uppermost layer. That is only the thing. This one is the uppermost layer. Okay. Now glycocalyx we saw. Now the lipids and proteins, this all red things, these are all integral proteins, and this one is the transmembrane proteins. Trans means only end. something end. Trans means end. This transmembrane at the side, it is at the most side. This is the sidemost, right? This is the sidemost. So, and these are all integral bodies. Integral means inside. Integral means inside. Inside something compact. But trans means totally outside. It's just attaching, not falling. But integral is compact within something. So integral proteins are inside, transmembrane is outside. Transmembrane is attached particularly to the membrane. But you can see here this yellow round objects, and this green grass like objects. These are called the phospholipid molecule. The this round objects and these grass like objects, they're all comprising of generally lipids. They're all comprising of lipids, but they have a charge. Remember, they have a charge, phosphate group. They have the phosphate group inside the phospholipid. Phospho means phosphate group, phosphate ion, PO4 minus, and lipid means lipid molecule, phospholipid molecule. If you combine it, it is mentioned phospholipid molecule. Now you can see two more terms. Carbohydrate side chain means just the thing that they've tried to make you understand. Single also that glycoprotein is a carbohydrate substance. It's forming the glycocalyx. Okay, this is the uppermost portion. This is the first layer. First layer. First layer. You can say this is the second layer. The second layer, and under it the submembrane cortical layer. There is no more function, just opening. Okay, just tubule. So in this picture it is not depicted. Under it, just third layer. So hydrophilic region and hydrophobic region. What is the meaning of what is the difference? This yellow things are outside, everything outside. In a cell, this is a cell. Outside there is water. Yes or no? Because our body is almost, let us consider human body. Our body is almost covered with water. If there is a cell inside our body, outside will be water. Okay. Inside there is cytoplasm. There is cytoplasm. That means the watery substance of the cell. So what will happen? Outside is also water, inside is also water. Hydrophilic. The word philic and the word phobic. Phobic means fear. Phobic means fear. Phobic means fear. Okay, fear. And hydrophilic. Philic means love, liking something. Philic means liking something. Phobic means fear. So here there is water outside the cell. Is the outermost, I said, glycocalyx. After that the other things inside is the cell, and below it also there is cytoplasm. Okay, so this region is called the hydrophilic region. This means there's yellow portions. These yellow portions, they are loving the water, the watery substance. So here is also water, and here it is also water. Cytoplasm is also watery substance, semi-fluid substance, so it is liking the water. But this region, this green grass like region, they are Generally, the tails of phospholipid molecule. We'll study about this main formula of plasma lemma. So, this is the hydrophobic region. They are away from the water. Okay, hydro means water, so they have fear from water. So they are away from the water. How? It is covered by this lipid molecules, the yellow portions. Hydrophilic region is covering it so that it doesn't get in contact with the water. Okay, that's the concept, nothing else. Okay, now formula of plasma lemma. Formula of plasma lemma. Phospholipid molecule. Okay, phospholipid molecule means glycerol. How? Phospholipid molecule is containing lipid molecule and phosphate group. Phosphate group, the sign is like this. I said you. PO4 minus. Phosphate group, the symbol is phosphate group, PO4 minus. Am I clear? PO4 minus. Now phospholipid molecule. This is the phospholipid molecule. As you can see here, phospholipid molecule. I think this is somewhat like this. It's too light, I think. Now I think it is clear. So phospholipid molecule. Phospholipid molecule means glycerol. How? You know that the plasma membrane is somewhat watery substance, which is possible because of glycerol. Okay, that only helps the fast transport because it is leaving very much leaving cell membrane. Cell membranes are always leaving, so it contains glycerol. And phospholipid molecule contains fatty acid plus fatty acid plus fatty acid means only the fats or lipids. So lipid molecule, let us consider two things, two fatty acids. So lipid molecule single one. Okay, so glycerol also has the same group, same formula, just the same. So we can consider the phospholipid molecule is equal to glycerol, or the plasma membrane can also be said that plasma lemma is made up of glycerol. So phospholipid molecule. Now this yellow portion, let me mark it with red. The yellow portion and this green portion, their functions are different. They are polar head. Polar head. Polar head. And this is called the tail. Okay, this is called the tail. It is also can be said the non-polar part of the phospholipid molecule. Non-polar, non-polar part also you can call it. Just actually Usually I am writing with not with stylus. I'm just using the, you can say, the pen or the ink shading, just so that is the problem. It is very hard to do with the also the laptop mouse. I'm sorry. And polar means it has charge. Polar and non-polar. I think you have studied in chemistry the types of molecular solids. There are also polar molecular solids, non-polar molecular solids, and hydrogen bonded molecular solids. So this is also the same, the types of solids like only. So the polar and non-polar. This may be positive, this may be negative, but it has charge. Some charge it has. But in case we are saying PO4 minus, so it is negative charge. It is the negative charge, not the positive charge. Okay, negative charge. It is constitutive of negative charge. This yellow portion is called the polar head. This is called the polar head. Okay, and this is called the tail or the non-polar part of the phospholipid molecule. So this yellow portion, this is consisting of the phosphate group, charge particles. Okay, I think you are clear. Now this is about the formula of plasma membrane. What is it consisting of? Main what? Now the cell wall. The cell wall we are talking about from very previous time. That what is cell wall? Now cell wall is the outer covering of eukaryotic cells. After plasma membrane, there is cell wall. Not in all cells. No prokaryotic cell has a cell wall. First thing. There is no cell wall in prokaryotic cells, and if there is cell wall in some bacteria or some prokaryotic cells, they have cell walls. That is consisting of heteropolysaccharide. You will see that in the homopolysaccharide. You will see that about the homopolysaccharide. Heteropolysaccharide means different types of carbohydrates. Different types of carbohydrates there, but here only one type of carbohydrate. Okay, so heteropolysaccharides, the prokaryotic cells, if they have, but eukaryotic cells, if they have, they must have homopolysaccharides. The cell wall must be made up of homopolysaccharide. And also there is a very unique fact that animal cells do not have cell walls. Their final is plasma membrane. That is the glycocalyx is the final. But here the plants, only plant cells and fungi cells have cell walls. So they are only having the cell walls. Okay, not animal cells. Eukaryotic cells have, but only plant cells and fungi cells, no other cells. It is made up of peptidoglycan. So peptidoglycan, we'll study about it later on. So what is it? I'm not telling you to just remind it what like other teachers say that it is made up of peptidoglycan. Just remember, it is not. You have to prove it. Otherwise, you will not understand. So it is made up of peptidoglycan. We'll study about it. So it is simple chain of homopolysaccharides. Homo means same. Homo means same. Okay. Homo means same. Homo means same. So it is a simple chain of homopolysaccharides, means same type of polysaccharide. Polysaccharide is a polysaccharides, many groups of saccharides. Okay, that is polysaccharide is a type of carbohydrate. Example, sugar, you can see C22H12O11, same thing. Sorry, I said wrong. C12H22O11. This is what? Polysaccharide. That means the sugar. C12H22 O11. Okay, and so it is combining of same type of carbohydrates, but prokaryotic cells, if they have different types of carbohydrates, and it is dead and thus rigid. It is very hard. It is very hard. It doesn't allow anything to come in. This type of things are called impermeable substances. Impermeable substances. Impermeable, impermeable substances. Impermeable substances. That means it doesn't allow, impermeable means it doesn't allow anything to pass through it. Like the drainage, we say it is permeable. The soil is very permeable. In less time of water, in less time, more water is flowing. That means it is more permeable. So like that only. Permeability means the passing of substances. Here it can be any material within the cells. So it is dead and it doesn't allow anything to pass through it. Okay. The comparison between plant and fungi cell wall. The plant cell wall and the fungi cell wall, they are different. Plant cell wall is made up of cellulose, which is a polymer of glucose. And if you say cellulose, then its monomer is glucose. Same thing. That's the opposite concept I say. And the fungi cell wall is made up of chitin. Chitin is a different substance. Will It is a chemical substance. No need to go into so much detail. So, and monomer is the N-acetylglucosamine. The monomer of chitin is the N-acetylglucosamine, or N-acetylglucosamine or the chitin is the polymer of N-acetylglucosamine. Just scientific terms. Plant cell wall, you see the pectin, the most amount is pectin. It makes the cell wall hard, very hard at the matured stage. And it also contains hemicellulose and the cellulose micropapille, that means through which the transport is occurring within the cell wall. What is that? Okay, we'll study about that. Hemicellulose, that's a scientific term. You don't need to concentrate on it. You can search it about in the internet. Middle lamella is the outermost part. We'll study about that. And cell wall, it is part of cell wall and plasma membrane. So plasma membrane, cell wall, and the outermost layer of cell wall is called the middle lamella. Cell wall has three types. Study about that. And fungi cell wall, you see the manoproteins, the proteins which are consisting, they are called the manoproteins. And the cell wall is there, and cell wall, and then the cell membrane, and then cell wall. Cell wall is, see the yellow portion is the cell wall, main part. That is the what? That is chitin. That is chitin. And there are the membrane proteins at the last. You can see two. They are depicted. They are the membrane proteins. Membrane is cell membrane. Plasma lemma is made up of sugars and proteins or lipids. Anything you can say. So membrane proteins there. So it is of the cell membrane. We saw the integral protein and the transmembrane protein. And beta glucans. That means this means beta, right? This means beta. This means beta. So beta glucans, they are the structural units or some type of proteins, you can say, a part of the cell wall of fungi cell wall. Okay. Now our next part is to prove plant cell wall is made up of peptidoglycan. I will come to it last. Let us complete it first. Parts of plant cell wall. Parts of plant cell wall: primary cell wall, secondary cell wall, and middle lamella. There are three types: primary cell wall, secondary cell wall, middle lamella. How will you depict? There are two cells. Here is the middle lamella. Here is the middle lamella, and this is suppose the cell wall. Draw it too large. Let this be the cell wall. So cell wall, this lining, the line, approx line, let it be the plasma lemma, plasma, plasma, plasma membrane. Let us write plasma membrane. So just after the middle lamella is the secondary cell wall. SCW. Let us write the type SCW. And this part just before plasma membrane, that is the primary cell wall. That is the primary cell wall. There is a primary cell wall. So the middle part, that's the dividing between the two cells, that is different, protecting the neighboring cells. You will see that is the middle lamella. And after this, if this is the middle lamella, then this is there is then this side there is secondary cell wall, and then there is primary cell wall, and then there is plasma membrane, that means glycocalyx. Then let us start from here. Middle lamella, secondary cell wall, primary cell wall, glycocalyx, lipid bilayer, submembrane cortical layer, and then the cytoplasm. Am I clear? Here also it will start same. This is the process. You may have to write the 6 different parts. It's the path through the middle, from middle lamella to cytoplasm, how it reaches. That's very interesting question. So primary cell wall, it is more prominent in growing plant cell. It is very helpful in growing plant cell because it is at that time it is somewhat light. It is not too much rigid. It is not too much hard. It is somewhat light. Many plant cells, when they are matured, it is more secondary cell wall is more prominent in still plant cells. That means when they are matured, still means matured. We are humans. I am still a kid, but still I am matured. So when I am matured, if I am matured, when I will grow bigger, when I will read in higher classes, so when I will do my college, that time I will mature. That time I will have secondary cell wall. Some plants or fungi plants, let us talk about, as it is written parts of plant cell wall. But let us talk about plant cell wall. So many plants, they only adopt the secondary cell wall, means they have the primary cell wall. They, with the situations, they change it or they recover it. They means just change it into secondary cell wall when they are matured. Still means my mind is now unstable. I am a kid, so means I'm thinking about that thing, I'm thinking about that thing, all here and there, what is happening. So this is unstable. When I will still, that means stable, that means the mature state, I will be not be thinking of anything else, but I will think of humanity, I will think of myself. That will be the still age. So primary cell wall is made up of cellulose, hemicellulose, and certain proteins. But secondary cell wall is made up of cellulose, hemicellulose, and lignin. That time lignin is also a chemical substance which make the cell wall thick. So that makes something thick. Primary cell wall is not too much thick. Lignin makes secondary cell wall something, and middle lamella, as it is protecting, it is rich in calcium pectate. Pectin, calcium pectate is consisting of pectin. What I said that time, pectin, pectin, pectin. It is consisting of pectin. So calcium pectate means the protein of pectin. So pectin is more harder than lignin. So middle lamella is too hard to protect the two cells, boundary of two cells, two plant cells, I should say, plant cells. It is present between the junction of walls of two cells. Okay, clear? Now, they have a very interesting question. If the cell walls are impermeable, we know that the cell walls are impermeable, nothing can pass through. Then how there will be transport of materials in the plant cells? How they will be transport, and transport of materials is very much required because the cytoplasm must be linked to the other cell's cytoplasm. Otherwise, the cell will be infunctional. There will the whole body will not function. Like here, the small cells will appear. There will no tissues. They will not combine. They will not form any tissues. So that will be a very big problem. And always remember that the transport of materials can only occur between cytoplasms. It cannot occur that mitochondria is sending some mitochondria is sending some mitochondria here. So it cannot happen that like that. So that is different material. So there are cytoplasmic bridges which are named as plasmodesmata. Plasmodesmata is a very important term. See, I will be back within some minute. It is very important. My battery is almost dead. So let me charge it and I will be back. Hello, I'm back. Just a few seconds I took. Um, Learned all this. Yes, we're talking about the plasmodesmata. So let me tell you something about the plasmodesmata. We were talking about that cell walls if they are impermeable. So how is the transport possible? Only when there is where only when there is two cytoplasms. There is the possibility of uh, transport of materials. Here is also cytoplasm here. Also cytoplasm. So only two cytoplasms can exchange the information because the cytoplasms have different materials or different organelles in them which release some of the proteins. Some of the proteins are present in cytoplasms also. Cytoplasm also there are some of the proteins and some of the organelles. Most of the things are combined of cytoplasm. Okay. Also, cytoplasm is a moisture type substance, semi-fluid substance, which helps to the transport of materials easily. Okay, so the plasmodesmata links the cytoplasm. This is very important. Cytoplasmic bridges, not any other thing. The plasmodesmata links. This is the plasmodesmata. It will only link two cytoplasms. It is not cell. It is cytoplasm. 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 It will link to cytoplasms. Okay, so it is impermeable, but there are some links between it. So it will help in their transfer of materials. So in this picture, you can see that this is the middle lamella. This is the middle lamella. As I discussed, two cells are there. Two cells are there. Two cells. Let us consider this part. The two cells. The two cells are combined here. Two cells are combined here. Same. So when they will combine two plant cells, cell wall is there, nothing will happen. So outer layer is middle lamella. Middle lamella is protecting. Outer layer is middle lamella. But here, this part, this part, this part, and this part, this part, and this part. There is middle lamella. But this part and this part. There is anything? Yes, there is a plasma membrane. Middle lamella, then secondary cell wall, then primary cell wall, then plasma membrane. As you can see here, this is the middle lamella, this is the middle lamella, then this part, this parts, this parts, this parts, these are the secondary cell wall. Okay, and this line, this I say it, that is very thin, it is not thick. Here in the picture it's shown that the middle lamella is very thick, so very thin, but actually it is the most thickest. Lignin makes thick, pectin also thick. Okay, so protection, both are almost equally thick. Okay, middle lamella, secondary cell wall, that's it, same thing. And here, this is the primary cell. Then you can have a question that, sir, you shouldn't call me sir, that why this portion, this portion is marked the primary cell wall, but I'm saying that this portion is the primary cell wall because we know that after plasma membrane the primary cell wall is there. It is not that anywhere we can put it, but it is also not wrong. In some cells, I said that biology is always something extensible or something differentiation or accept. The word accept is very used in biology, that everything is this, but except this. There is biology is only considered the science of exceptions. So there can be exception in this picture also. Okay, can be placed there also because it is the growing state where more we do not know. These are the cell organelles. There is cytoplasm. These are also the cell organelles. Some cell organelles. This can be considered the nucleus, nucleolus. It can be considered. So this is the concept. Now we left one slide, I think. Yes, this slide. To prove cell wall is made up of peptidoglycan, at the not the beginning of the class, at the middle of the class, I said that cell wall in this slide, I think. Now this slide, yes, it is made up of peptidoglycan. So how it is made up of peptidoglycan? Why will say it is made up of peptidoglycan? What is peptidoglycan? These questions are very common. So to prove that, I have prepared this slide. This will not get anywhere. This is not required. Also, no one will ask you, just to explain you, or these are my questions, you can say. So this is very interesting question. To prove that plant cell wall is made up of peptidoglycan, how is it possible? So to say that, I will explain you this. Now in the proof you can say that, let us consider the endoplasmic reticulum. We have, I have not given any animal cell, plant cell diagrams because that are almost in all books they are given. So I have not given again and again. It's useless. This may be the nucleus. This is the surrounding plasma membrane. These things are called the ER or the endoplasmic reticulum. This is also called ER. ER. This is also called ER. So here, rough endoplasmic reticulum, smooth endoplasmic reticulum. RER. ER has two types. RER. RER and SER. RER and SER. They are short forms of rough endoplasmic reticulum, smooth endoplasmic reticulum. Some ER have, sorry, I did a mistake. Suppose this is a ER. This is the RER. This is the RER. You can say. And these are called ribosome. These are called ribosome. Ribosome. Actually, again, I am saying that I am writing with this computer pen. That's why everything is going all worst. Handwriting is going too worst. I know it. But main thing is you understand the concept. This is very important concept. Ribosomes are present. These are the ribosomes. These are the ribosomes. These are also called the protein factories of the cell. They prepare the proteins. And suppose your mom, your mom is preparing food, okay, in your home. Suppose nowadays many people have many things to do, so they do not get time. But for the timing, let us consider that your mom prepares food in home. If your mom prepares home and food, you and your father doesn't you eat the food? Let us consider the whole RER as home. RER is rough endoplasmic reticulum RER. So it is rough because of the presence of ribosomes. It makes the rough surface rough. So smooth endoplasmic reticulum, that is smooth. RER is ribosome is not present. That's why it is smooth. So RER. Your house is RER. Your home is RER. Happiness and love makes a house a home. You know this. Let us consider not consider that. Let us consider home. RER is your home. Your mother is the ribosome. She's preparing the food. Will you and your father not eat? So the whole RER will get the benefits of ribosome. What the ribosome will do, if ribosome is called the protein factories, RER can also be said that it produces proteins. It can be said because it contains ribosomes. Okay. A smooth endoplasmic reticulum produces lipids or fats. Now, as lipids and carbohydrates almost belong to the same group of nutrients, we can consider that the ACR produces sugars or carbohydrates. That means lipids and carbohydrates. While reading the nutrients, almost in class 6 or 7, we have studied that fats and carbohydrates, proteins, vitamins and minerals, water, roughage, and minerals. Why we say fats and carbohydrates and vitamins and minerals? Why we do not say proteins and minerals, vitamins and lipids? Why we do not? There is no link. Vitamins and lipids, their functions are almost same. That's why we say that carbohydrates and fats, they help in energy production, and vitamins and minerals fighting germs, producing immunity, and proteins are building bodybuilding foods. They help the body to be stable. So fats and carbohydrates are almost in the same group because they almost perform the same function. There may be some difference, but almost the same. So let us consider that SCR produces not lipids but sugars to prove it. Okay, and sugar, I again am saying sugar is a, let it be, sugar is a example of sugar, glucose, sugar, and also sugar is a polysaccharide which is a carbohydrate. Okay, SCR produces sugars. Also, you can say for that timing I said remember that glycocalyx. Glycocalyx it is consists carbohydrates. So cell wall, glycocalyx, the last layer of plasma membrane. All cells have plasma membrane, and upper that some cells they are having cell walls. The plant have cell wall. Below it is plant plasma membrane. Plasma membrane last layer glycocalyx that contains carbohydrates. You can also prove like this, or you can say that the sum of the carbohydrates are enwrapped in this cell wall for the making of the cell wall. And the way this outer membranes are made, This is called membrane biogenesis. We write the term. It is the membrane biogenesis. Membrane, membrane biogenesis. Very highly I have written. Membrane biogenesis. Membrane biogenesis is the making of the outer membrane of any cell with the help of proteins and sugars or lipids produced by the endoplasmic reticulum. It has parts, so let us consider the whole endoplasm. So we know that the proteins, same thing. That this process is called membrane biogenesis. Okay, let's try explain it. We know that the proteins and sugars from the ER make out the outer membrane, which is cell wall in plant cell. In plant cell, not only cell wall, not only plasma membrane is made. Plasma membrane is also made. Plasma membrane in every cells. They are made of proteins and lipids or sugars. In many books, you will get proteins and lipids, but you can consider sugars also because I say they belong almost to the same group. They produce energy, starch, rice. You get energy. Also butter, ghee, oil. You get energy. So the the proteins and the proteins and the sugars. Let us consider again. I am saying outer membrane. Also, plasma membrane is made in plant cells. Also, cell wall. Also, the membranes, outer membrane, not the nuclear membrane. For that, I have written outer membrane. Outer both membranes, two membranes. This cell wall and cell membrane, but this case let us consider cell wall in plants. So, by the process of membrane biogenesis. Therefore, cell wall is equal to proteins plus sugars. Cell wall is made by the proteins and sugars. It's same thing from the ER. So, proteins plus sugars. Proteins have mainly two types. They are combined, or they are a form of combined amino acid chains. So, they have two forms: peptones and peptoses. Actually, but today's form we can also say peptices. The full form. Full form I'm saying plural form. You can say that peptones and peptides. Sugars. One example is glucose, the main component of plants. Cellulose. Cellulose is a polymer of glucose. Let us like the monomer, that is glucose. Sugar, we have derived glucose. And proteins, peptones and peptides. Two types mainly. They can be broken. Proteins can be broken into peptones and peptides, while in the stomach, the proteins are broken into peptones and peptides. While reading digestive system in class 7, I think you have read. So peptido, how we had from peptones and peptides, pept. From that word, we are taking pepti common. Pepti, we are taking common. Do means two. Do means two. Do means two. I've written here two proteins. Peptido. Pepti we are taking common from peptones and peptices. From peptones and peptices we are taking pepticommon. Pepticommon. And do as two things, peptones and peptices. Two things. So two proteins. Let us consider them two different types of protein. Two proteins that are divided from proteins. So two proteins, do pepticommon, so peptido formed, and from glucose we are taking glycan, like you know, prokaryotes, carry on from nuclear from carrion nucleus is derived. Carrion means something centrally placed. That is nucleus. Okay, carrion can be derived from nucleus, or nucleus can be derived from carrion word. So glucose can be derived from glycan, or glycan can be derived from glucose. Just simple language, or you can say Latin form. Glucose, a scientific term, glycan, all both are same. To prove, we are showing it that so peptido plus glycan, they form peptidoglycan. So cell wall is made up of peptidoglycan. We have proved it. If you have any confusion, you may watch the video again, and if you like it, please like it in YouTube, and please subscribe it. If you like it, please like it in YouTube, and please subscribe it. I hope you would like it, and from the next time I would be coming with better slides, many other topics of biology. We can continue with the cell or different subjects: history, geography, physics, chemistry, maths, English, any topic I can come with the next video. If you would like it, I would be more encouraged and produce more good teaching processes. Thank you, and very much thank you for watching this video. That's it. Thank you. I hope you would like it.